Hello everyone, welcome back to the session. So in the last class we have discussed about the short answer questions in the diversity of living world. Okay, so that is in the first year. So in today's class we are going to discuss about the short answer questions in the second year and the chapter called the digestion and the absorption. Okay, so this is the 9th day of our class, right? Okay, without any delay we'll enter into the session. So the first important question will be the draw a neat labeled diagram of the LS of a tooth. Okay, so it just this is not an exact picture, but there is a difference. Even though you are not able to draw the exact picture, but you can draw a clear, distinct picture with a proper parts marking. So that is an important thing. Okay, so this is just a rough diagram, but see, look at this picture. This is very neat. Okay, so just follow this picture and write it. Okay, so this is the LS of a tooth. So in the tooth we they are short round like structure which is considered as a bone. Okay, so this is considered as a root canal. Okay, so this is the root canal. So if there is any the tooth is not functioning properly and it is we have to, if the enamel is completely eroded, there comes the root canal treatment. Okay, so this is what a root canal area, and this is gum. Okay, so in each and every tooth there is a structure called as a gum, and this is responsible for the loss of this bleeding. Okay, so vitamin C is if there is no proper vitamin C, it leads to bleeding gum and loosening of the teeth. Okay, so this is a pulp cavity. So this is a cavity, this is considered as a pulp cavity. Okay, so and this is considered as an enamel, the most important point. Okay, so most important layer which is covering the teeth, which is considered as an enamel. Okay, this structure is called as a dentin, which is exactly the tooth structure. Okay, so this dentin is covered by another layer. This is called as an enamel. So this is an important question. Just make a note of it, and this diagram is very important. Okay, so this structure, this clear structure with the proper parts marking is where it's sufficient to score marks. Okay. Now, so if you find any questions like this, try to approach this question. Why? Because you can easily gain the marks. Okay, so instead of a question with a lot of theory part, if you just draw the diagram and label the part, if you label the parts, you can get a maximum marks. Okay, so now the second question will be the describe the process of digestion of proteins in the stomach. Okay, so gastric juice secretion. Okay, so first you should know a digestion. What is mean by digestion? A complex structure. Okay, a complex structure which is converted into a simpler molecules. Okay, a complex structure. Whenever we are eating the rice, whenever we are eating the fruits, whenever we are eating any pulses variety, cereals variety, so all these are considered as a complex biomolecules. Okay, so whenever as a result of this digestion process, so if all these complex molecules enter into our stomach, straight away it can't be absorbed by all the cells. Okay, so that is why this complex molecule has to break down first. Okay, then it has to convert it into a simpler molecules. Only then it is easier for the absorption process. So you should know, for cells to absorb all the essential nutrients, the proper digestion has to take place. Okay, so only after the digestion that all the cells has to be absorbed all the essential nutrients. Okay, so you should know the digestion process. So if it is a carbohydrates, this is a polysaccharide. So this is nothing but that complex molecules. Okay, so this is a carbohydrates, polysaccharides, that will be converted into a monosaccharides, that is glucose. Okay, so if it is a protein, it will be converted into an amino acids. Okay, so if it is a lipids, it will be converted into a fatty acids plus glycerol and fatty acids. So these are all the monomers and these are all the polymers, polymers, right? So this is what a complex molecules, it is converted into a simpler molecules, and this process is called as a digestion process. Okay, and you should know, whenever I'm saying the digestion, immediately after that you could remember the organ stomach. Okay, so it is a misconception that stomach is involving in most of the digestion. It is wrong. Okay, only small intestine is responsible for most of the digestion process. Okay, so now first thing, so how the complex molecules is getting converted into a simpler molecules. It is very simple, just by secreting the gastric juice. Okay, just by secreting the juice. If it is a stomach, we are calling them as a gastric juice. If it is a pancreas, we are saying that as a pancreatic juice. Okay, if it is an intestine, we are saying that as an intestinal juice. All these juices contains an enzymes which is responsible for breaking the complex molecules into the simple molecules. Right? Now the first thing will be the gastric juice secretion. So the gastric glands, always the secretion is done by some glands. If is that, as this is a gastric juice secretion, definitely it will be related to the gastric glands. Okay, so the gastric glands secrete the HCl, that is hydrochloric acid. Prorenin means what? Renin is a protein, and which is in this renin is an inactive form, which means inactive renin. Pepsinogen, pepsin is an active protein and pepsinogen is an inactive protein, and the bicarbonates. Okay, so all these juice contains an inactive enzymes. How it is getting activated? It is getting activated by some sort of enzymes. Okay, so the pepsinogen, it is converting into a pepsin, okay, by the hydrochloric acid, which means you should know that this pepsin can survive in an acidic environment. Okay, and we know that this hydrochloric acid, this is an acid, which means they are having a low pH. Okay, so this pepsin and renin, it is very it becomes active only when it is meets an acidic environment. Right? Now the action, so we have studied how the enzyme is getting secreted in the gastric juice, how it is getting activated in the acidic environment by the hydrochloric acid, but how the action of what will be the mechanism of that particular enzymes. Okay, so the renin in case of infants, okay, this renin is present in the infants, that acts on the casein. Casein is considered as a milk protein. We know that most of the baby will depend only on the milk. Okay, so initially up to the 6 months to 1 year, all the baby will depend on the milk. Okay, so they have to digest the milk. Okay, so they have to digest the milk, so that they are having a casein protein. Okay, which means this renin. See, here it is not renin. R-E-N-I-N. We are having two renin. Okay, R-E-N-I-N, R-E-N-N-I-N. This R-E-N-N-I-N is somewhat related to the milk protein and the enzyme which is responsible for digesting the milk, and this renin is responsible for, it is involving in the kidney function. Okay, don't get confused with these two things. Right? So now this renin, which is present in the infants, why? Because it is essential for the milk digestion. It is acting on the casein. Casein is nothing but a milk protein. The protein which is present in the milk is called as a casein, and the casein is getting digested only because of the presence of the protein called the renin. Okay, so in the presence of the calcium. So for this digestion, calcium is also very, very important. Together they are forming the calcium paracaseinate. That is nothing but a curd. Okay, see, the baby is intaking the whenever the baby is intaking the milk, it is like in the liquid form, milk form. Okay, so whenever the renin is acting on that particular casein protein, it turns like a curd, that is calcium paracaseinate. Okay, so now this pepsin, what is the function of this pepsin? Pepsin is acting on the protein. Okay, pepsin is acting on the protein. Okay, now this protein is converting into proteoses and the peptones. Okay, proteoses plus peptones. Okay, so now this calcium paracaseinate is getting converted into the peptones. Okay, so this is what an action of an enzymes. Okay, this is what an action of an enzymes. In the protection of the stomach lining, the bicarbonates which is associated with this mucus, which is forming a barrier to prevent a damage from the HCl, which is hydrochloric acid. See, whenever we are feeling hungry, the acid which is present in our stomach, which is nothing but an hydrochloric acid, it is continue to digest all the mucus layer which is present in the digestive tract. Okay, so because of this acidic environment, so that is why how it is getting protected with this stomach lining. We know that HCl is considered as an acid. Okay, HCl is an acid and that has to be neutralized by base. Okay, when it is mixed with the base, it is getting neutralized, and that base is nothing but a bicarbonates. Okay, so the bicarbonates plus the mucus form a barrier. Okay, there will be no mucus lining. The HCl can easily erode the gut lining, so that is why there is a proper covering which is called as a mucus, right? Now, okay, so that's all about this question. This is what a process of digestion of proteins in the stomach. We are not seeing the entire part of digestion. Okay, how the carbohydrates is getting digested, how the protein is getting digested, how the lipid is getting digested, everything will be the different phenomena. Okay, so we just saw that the digestion of the proteins. Okay, now the third question will be, if you take a butter in your food, how does it get digested and absorbed in our body? Explain. So butter is considered as what? It is coming under carbohydrates or lipids or proteins. We are going to see that. Okay. So how it is going to take? Initially, all the things will be happening in the mouth. We are taking in the mouth. See how the alimentary canal will be. See, it is mouth. The food has been taken into the mouth. It has to reach the pharynx. Okay, through the esophagus, it is reaching the stomach, and from the stomach it is entering into the small intestine, and from the small intestine it is entering into the large intestine, and from the large intestine it is to the rectum and the anus. This is what a proper alimentary canal. Now the food has been entered into the mouth. Definitely the enzyme is secreting in the mouth. Okay, so that is called as a salivary amylase. In case of carbohydrates, this salivary amylase is involving in the digestion. As this is a butter, the lingual lipase. Okay, lingual lipase. Lipase is nothing but a lipid, a fat molecule to digest the fats. Okay, what will be the action if we take the butter initially? See, we are taking the butter in our mouth. Which enzyme is getting activated? Lipase is getting activated. Now, what this lipase is going to do on a butter? So, hydrolyzes a small fraction of the triglycerides, and this triglyceride, so this lipid, we know that is a triglyceride, and this triglyceride is converting into a diglyceride plus the fatty acids. Okay, so it is not entirely, so as I said before, if it is a fat molecule, it has to be converted into glycerol plus fatty acids, but initially this tri has been converted into di. It is a minor digestion that has been taking place in the mouth. Okay, don't get confused with that. So always 30% of digestion has been taking place in the mouth. Okay, where it will be the site, that is in the oral cavity, minor digestion is taking place. Okay. Now, what is in case of the stomach? So the pharynx and esophagus is considered as just a pathway of that food. Okay, so the stomach, what will be the name of an enzyme? That is nothing but a gastric lipase. Okay, so if it is present in the mouth, we are calling them as the lingual lipase. If it is present in the stomach, we are calling them as a gastric lipase. Okay, what will be the action of this gastric lipase on this diglyceride plus fatty acids? See, the action, this partially digested triglycerides, which is converting into diglycerides plus the fatty acids. This is what taking place in the stomach. Not again, here also a digestion is taking place partially, not completely. Okay. Now, what if in case of this small intestine? In small intestine, where exactly? So you should know this duodenum, ileum and jejunum. Okay, duodenum, ileum and jejunum, all these were the parts of this small intestine. Okay, now this bile salts. Bile salts, very important. Bile salts is involving in the emulsification of fat, which means digestion of fats. Okay, so this bile salts emulsifying the fat, increasing the surface area for all the enzymes, which means here the enzymes will be the pancreatic lipase. Okay, pancreatic lipase. What will be the action of this particular pancreatic lipase? This is the one hydrolyzes the triglycerides, that is converting into monoglycerides plus 2 fatty acids. So this is where the final digestion has been taking place. Okay, so now all the food has been digested, where it has to be absorbed. Okay, so it has to be absorbed by all the cells of our body. Okay, how it is getting absorbed? So what will be the process? Here the monoglycerides and the fatty acids form the micelles. Okay, which means it a round like structure. Okay, together with this bile salts, all these monoglycerides and the fatty acids, it is forming the micelle like structure. Due to the, and it has to be all these together, this micelle, it is called as a micelle, which is diffused into the intestinal cells. And what is called that intestinal cells? That is called as an enterocytes. Okay, so inside this enterocytes re-esterified to the triglycerides. So this is very important. Okay, so that has to be digested first. After that, once again inside the enterocytes, it has been re-esterified to the triglycerides back. Okay, so all these triglycerides has been packed into the chylomicrons. Another important point, chylomicrons. Okay, so now, so the lipid has been transported to all the cells of the body in the form of this chylomicrons. Okay, how it is getting transported? See, this chylomicrons, it is not entering the blood. Before it has to be enter into the blood, it is entering the lymph. Okay, so after that it will be enter into the blood for storage or energy. Okay, so this is what happening in the absorption process. Okay, so this is a simplified equation. Okay, so this emulsified fats, okay, so in the presence of these lipases, this fats has been converted into diglycerides. Again, in the presence of this lipase enzyme, it has been converted into the monoglycerides. Okay, so if the fats in the presence of this bile salts, it has been converted into the emulsified fats. Okay, and monoglycerides in the presence of this lipase, it has been converted into the fatty acids and the glycerol. Okay, so this is what exactly happening in the digestion of this lipid. Okay, so they directly didn't give the word lipid, but instead they have been given the word butter. But you should know butter is the fat. Okay, so that's all about this third question. So the fourth question will be this: what are all the functions of this liver? Okay, what will be the functions of this liver? The first thing will be the bile secretion. So whenever we have seen this third question, we have come across with the word this bile salts. Okay, so this bile salts is secreted by, it is present in the bile. This bile is nothing but secreted by this liver. Okay, so the liver is secreting the bile. Now what is the function of this bile? The bile secretion contains this bile salts. Okay, so in this bile salts, what is present? Glycocolate, taurocolate, and the pigments, very important. So these are all the things which is called as a bile salts, and another thing will be the bile pigments. Both will be present in this bile only. Okay, so the pigments like bilirubin and biliverdin. Okay, so that is why this gives an yellow color. The pigment is, we are saying this bilirubin and biliverdin is a bile pigment, and bile pigment definitely needs the color. Okay, and this pigment has an yellow color. So that is why the human waste excretory matter is look like this color. Okay, so this is an important thing. The first thing will be the bile secretion, and the second one will be the carbohydrate metabolism. What is a carbohydrate metabolism? Very important. glycogenesis, glycogenolysis, gluconeogenesis and lipogenesis. Okay, so glycogenesis means what? This is what glycogen. Okay, glycogen, genesis in the sense synthesis. Okay, so glycogen synthesis. Okay, so next the glycogenolysis, which lysis means break. Glycogen is getting breaking. Okay, glycogen is breaking. Okay, so that is what glycogenolysis. Gluconeogenesis, which means gluco means glucose. Okay, glucose, neo means new. Okay, genesis means synthesis. Glucose is synthesizing from the newer sources. This is what gluconeogenesis. Okay, lipogenesis in the sense, genesis means synthesis, lipo means lipid, lipogenesis. Okay, so all these were the carbohydrate metabolism which is happening in the liver. Next, lipid metabolism. Okay, so the lipid metabolism, they are synthesizing the cholesterol and produces the triglycerides. Okay, so they are producing the cholesterol. They are synthesizing the cholesterol as well as they are producing the TGL, that is triglycerides. Okay, and protein metabolism. So these are all the energy product only, right? Now the protein metabolism. The protein metabolism, they deaminates amino acids and converts ammonia into the urea through the ornithine cycle. Okay, see, deaminates amino acids and converts this ammonia into the urea through the ornithine cycle. See, you have to know that humans are excreting the urea in their excretory product. We can call them as the ureotelic. So we are ureotenic animal because we are excreting the urea in the excretory matter. Okay. So before that, they have to convert. So actually, this is the one waste product that has to be eliminated, as this is very toxic and needs more water to eliminate from the body. Okay, so this ammonia has been converted into the urea. Okay, and that particular cycle is known as an ornithine cycle. Okay, and this ornithine cycle is taking place in the liver. Okay, so this is an important thing. Okay, and the fifth important thing will be the Cori cycle. Okay, so this Cori cycle, so you should know it is converting the lactic acid from the muscles into the glycogen. Okay, so this is an important phenomena. What is the concept behind this Cori cycle? Whenever we are doing the exercise. Okay, so whenever we are doing the exercise, our cells are devoid of this oxygen. Okay, our cells are in need of more oxygen. Once the muscle is exhausted with this oxygen, so all the cells are respire anaerobically. See, I repeat. See, whenever we are doing a normal regular work, we are having a sufficient oxygen. Whenever we are doing an exercise, whenever we are in a hurry, okay, our muscle cells are in need of more oxygen. In the vigorous exercise, our muscle cells are exhausted. Oxygen is getting exhausted in the muscle cells. Okay, so at that time, as there is no oxygen, immediately after that the cells have been shifted to the anaerobic respiration. Okay, so that is why it is getting converting this lactic acid from the muscles. Glucose is getting converted into the lactic acid and it has to be converting into the glycogen. Okay, so this cycle is called as the Cori cycle. Okay, now another important thing will be the detoxification. Okay, detoxification means what? It neutralizes the toxic substances entering from the gut. Okay, see, so we are having an endoplasmic reticulum. Okay, in the endoplasmic reticulum, we are having a rough endoplasmic reticulum and we are having a smooth endoplasmic reticulum. Okay, so in the rough endoplasmic reticulum, if the endoplasmic reticulum have ribosomes, we are calling them as the rough endoplasmic reticulum. If the endoplasmic reticulum not having any ribosomes, we are saying that as a smooth endoplasmic reticulum. The functions of rough endoplasmic reticulum is the synthesis of ribosomes. Okay, the functions of the smooth endoplasmic reticulum is the synthesis of lipids and the detoxification process. Okay, so the smooth endoplasmic reticulum concentration will be higher in case of this liver cells. Liver cells is nothing but this hepatic cells. Okay, hepatic cells. Okay, so in this process, this neutralizes the toxic substances entering from the gut. Okay, how liver is involving in the detoxification process just because of the smooth endoplasmic reticulum. The concentration of smooth endoplasmic reticulum will be higher in case of liver. So the next point will be the thermoregulation. Okay, so the thermoregulation here it will be participates in heat production due to the glucose metabolism. Okay, see whatever be the process happening inside the cell. Okay, whatever be the process will be, the side product will be carbon dioxide and heat. Okay, so that will be mostly heat. Whatever be the mechanism, either it will be the catabolism or anabolism. See, catabolism in the sense complex molecule has been getting converted into the simple molecule. Complex molecules is getting converted into the simple molecules. In case of anabolism, many simple molecules together form the complex molecules, and this is considered as a metabolism. Okay, so as whenever the metabolism is happening, okay, whenever the metabolism is taking place, the side product released will be the heat. Okay, the byproduct will be the heat. Okay, so that is participating in the heat production due to the glucose metabolism. So in the glucose is getting metabolized. So we have studied the carbohydrate metabolism, that is nothing but a glucose metabolism that is happening in the liver. So it is releasing more amount of heat. So that is why it is responsible for this thermoregulation process. And the 8th important point will be the hematopoiesis and erythroclasts. Okay, so this hematopoiesis is nothing but hemato, in the sense all the blood cells is getting synthesized. It is acting as the hematopoietic organ. Okay, it is acting as the hematopoietic organ in the fetus. In the fetus, not in the adult. Okay, so in the fetus it is considered as a hematopoietic organ, which means all the blood cells, most of the blood cells will be synthesized in the liver. Erythroclastic organ in case of the adult. Okay, so this is the most important point, just make a note of that. Okay, and the 9th important point will be the plasma protein synthesis. And this plasma protein synthesis means what? Plasma proteins. So in the blood we are having two important things. Okay, so in the blood we are having two important things. One will be the plasma, and another one will be the formed elements. Okay, so in the plasma we are having water, electrolytes, solutes, and the blood proteins. Okay, so this formed elements are they are having the blood cells, that is RBC, WBC, and the platelets. Okay, so this plasma protein, they are nothing but this globulin, albumin, and the fibrinogen. Okay, so globulin is responsible for this antibody pressure, that is immunoglobulins. Okay, so albumin is responsible for the osmotic pressure, osmotic balance. And fibrinogen is involving in the clotting functions. Okay, so all these plasma proteins is getting synthesized in the liver. Right, now phagocytosis, another important thing. So in the WBC, we are having 5 important cells: neutrophils, eosinophils, basophils, lymphocytes, and monocytes. So this monocytes, whenever it is streaming in the blood, we are calling them as the monocytes. Whenever it is reaching any tissues, we are calling them as the macrophages. Okay, so this macrophages is involving in the phagocytosis process. Phagocytosis in the sense it is engulfing the microorganisms, okay, and digesting them. Okay, so this macrophages, if it is present in the liver, we are calling them as the Kupffer cells or Kupffer cells. Okay, so if it is present in the lungs, it is called as an alveolar macrophages. Okay, so if it is present in the brain, we are calling them as the microglial cells. Okay, glial cells. So this Kupffer cells is nothing but a macrophages which is responsible for the functions of phagocytosis process. This is the one removing the microbes and the unwanted substances. Okay, so these are all the questions which are all very important in the digestion and absorption of lesson. Okay, so in tomorrow's class we'll meet up with you. I'll meet you in the next chapter. Okay, thank you so much.